Hello, everyone. We are still waiting for attendees to join us. We have a lot of attendees coming, and I'm very pleased to see that's from all over the world. We are represented by all the ART societies and also many from my previous work area, the Middle East, and also from down under, my second home, and South Africa, of course. Thank you for your patience. If we are about to address this, we have still many coming in. I think I start. So, hello everyone, and for joining us today's webinar, Airborne Awareness. My name is Lars Johansson, and I will be your moderator. I've been working for 30 plus years in this ART business. I worked as clinical embryologist, lab director, but also director for the embryology part in Cooper Surgical. In that position, I went and saw a lot of clinics and the problem they have and helped them to optimize the results. And I also put up the training schools in Mumbai and in Copenhagen, Denmark. My background is that I got my PhD in developmental biology in 1988, where I looked into the effects of chronic exposure for male mice and looked at their sperm chromatin and how it affected the actual development of the embryo, blastocyst, and implantation. And during that period, I was also responsible for workshops and dissections, etc., but also equipment. And it taught me a lot about what not to use and how to modify them. In the year after 1989, I got a scholarship to go to Australia, Monash, Center for Early Human Development, under supervision of Ellen Transon. And we did a lot of funny stuff. And I was actually selecting sperm according to their motility pattern for Susie at that time. And we were doing a lot of other things and injecting sperm heads, and nothing happened, as you know, by now. And throughout my career, I've been pursuing, trying to help to improve outcomes, either by the building material, design, selection of disposables, equipment, and of course, how you work inside the lab, and especially the environment. So, and of course, other things I've done is actually, I've been involved with a lot of different research, design of equipment, as well as coming up with new ideas outside the box, so to speak. I'm working now as a consultant, auditor, lecturer, and mentor for both clinics and upcoming companies. Today's webinar, we will be discussing the impact of transformational air purification system towards improved patient care in IVF and healthcare, unique beyond its genesis. During this presentation, you can also send in questions to the QA panel, and those questions we will answer at the end of the session. I'd like now to introduce our speaker, Dr. Kathryn Worrilow, the founder and CEO of LifeAire Systems. She received her doctorate in anatomy and cell physiology from the University of Virginia School of Medicine and completed her postdoc fellowship in reproductive biology, physiology, and infertility at the University of Pennsylvania School of Medicine. She has worked in ART clinics for over 20 years and as a scientific director. And she also been interested in the interaction between sperm and eggs, and of course, how that also affects the embryo development from the paternal side. But she also, as you understand, looked a lot into the impact of the ambient air and how you get better results and outcomes. She also developed and designed, together with colleagues, the LifeAire system. That's patented air purification technology, and the system and air IVF is actually deliver a very high quality, clean air for the IVF environment by installing it in the induct system. So it's not a standalone unit. It's been installed in 50 to 60 installations so far, air clinics worldwide, and it's just increasing. She has authorized 60 papers, chapters. She has been invited as editor, reviewer, speaker, and sits in many different boards. In addition, I would say she has been given awards from many other places, and one of the recent one is actually from the Pennsylvania governor, and because of the help the ventilation did through this latest pandemic, but also to the recovery of the economy. So this system, which you will see, it actually reduces and eliminates healthcare acquired infections and airborne pathogens. She's also, and I guess there are some others here, you're co-author of this book, where you can actually read a lot about the clean room technology and what you can do with it. And I therefore invite Katie to take over and start the show.
Katie. All right. Thank you, Lars, and good afternoon to each of you. We thank you for joining us today to discuss, really we wanted to share the pearls and take-home messages from well over 20 years of research specific to the IVF culture environment and how these learnings really defined the airborne metrics critical for the successful and consistent culture of the human embryo, and ultimately led to the design of an air purification technology to deliver these defined metrics. Now, I'd like to share these learnings with you as well as some of the cellular and clinical data resulting from the use of the technology. Now, as many of you, I served as an IVF laboratory director, as Lars shared, helping individuals and couples fulfill their dreams of having a family. Throughout the 20 plus years in doing so, our team was critically focused on reducing variables that threaten the success of our cell culture environment. As many of you, we tried to reduce, control, eliminate as many variables as we could to deliver the highest clinical pregnancy rates and therefore level of care to our patients. The variable that we're going to discuss today is one that was quite elusive to us, extremely dynamic, and offered a negative impact on our outcomes. The ambient air, the air that serves your IVF laboratory, your clinical procedure rooms, your ante room that surrounds all of our IVF procedures. In the year 2000, we were asked to design and build a new IVF program and laboratory. So we obviously didn't know then what we know now, but assuming that air quality was a variable that was impactful to our process and wanting to remove that as a negative influence, we designed and built a Class 100 or ISO 5 clean room from the ground up, assuming that would remove or control the variable of air. It was a space, it was an unbelievable laboratory. Again, Class 100 design, we had a dedicated air handler, a sealed plenum, 30 air changes per hour, obviously positive pressure, live monitoring. Now, we didn't know at the time, but this was going to be truly the gem of this laboratory design, live VOC monitoring. Laminar flow, we had 450 pounds of molecular media, carbon, and potassium permanganate. We had ultra-final filters. We had UVC exposure on all of the ultra filters as a final point of filtration. Airflow over all critical points of process, and we followed clean room SOPs. But again, you will learn as I share some of our own take-home messages how critical this live monitoring was to the process. So how effective was this space? Were we successful in removing the variable of air? Fast forward 10 years, this slide shows you 3 years of data. The short answer is no. There were times where we exceeded the average clinical pregnancy rate in the United States by 10 to 15 to 20 percentage points and then repeated it. But then in a third cycle, we dropped down to 16% clinical pregnancy rate across all maternal ages. Fast forward 10 years, what we learned is that outside activities outside of our laboratory on the hospital campus, it was a very large progressive hospital, activities that were happening outside of our laboratory were generating very low levels of airborne pathogens, which were coming through our HVAC system and entering the clean room. For example, in this first dip, after we went back through all the live monitoring data and linked it with activities that were outside the laboratory as well as inside our laboratory, just typical SOPs, we found parts per billion toluene and low levels of bacillus. Bacillus is a spore associated with soil. There was a large construction project ongoing at that time, as well as they were resurfacing the medevac pad. Resurfacing with asphalt, fresh asphalt produces embryotoxic toluene, but it wasn't a large amount of airborne pathogens. It was literally parts per billion and very low levels of viable particulates or your bacteria, fungi, or viruses. Our second drop was also associated with construction, producing aspergillus, a fungal spore, and aldehydes within our space. So what the live monitoring of the clean room actually provided was a roadmap to help us determine what level of VOCs consistently are critical for the successful growth of the human embryo. What level of viable particulates, your bacteria, viruses, and fungi, what levels are truly critical? What levels are necessary? So once we determined what airborne metrics were critical, we sought to purchase a system that would deliver this. And much to our surprise, there was nothing commercially available that we could purchase to deliver the air that we now knew was so important. And that is really what led to the genesis of the LifeAire systems. So we pulled together carbon chemists, chemical engineers, clean room engineers, nuclear physicists, and basically worked backwards and said, this is the air that we know we need in our IVF laboratory to have consistent support to completely purify all outside source air. As we'll share in this, in the next few slides, you can never control your neighbors. You can't control construction schedules when they resurface a road or a rooftop. And that's all going to be activities that are ongoing outside of your laboratory. So we wanted a system that no matter what it was delivered, would purify the outside source air and all recirculated air within the laboratory, because we generate a lot just by our staff and our SOPs. We wanted all of that air to meet the metrics that were defined by the 10-year study. So the system was designed, patented, tested for years, and then launched and is now in place in a number of leading medical centers and IVF programs throughout the US, and as well as we have our first in Europe at King's Fertility in the UK, our first in Canada, and several in China. But they've become really lifelong partners and clinical providers in sharing their data with us. Mayo Clinic, Stanford, Northwestern, University of Connecticut, Wake, Iowa, Yale, Duke, UCSF. And just as I said, just ongoing clinical partners in sharing their data. Our goal with the design of this system was pretty simple. We wanted to take the typical level of VOCs, and this is the typical level pre-installation of many of our first systems. We wanted to take total VOCs to below detection. We wanted to drive this variable to 0. We also wanted to drive the variable of viable particulates, or your bacteria, viruses, and fungi, as well to below detection. But what's happened as a result of that, in truly removing this singular variable, and as we all know, there are 1,000 variables in human IVF, but just by controlling one, no matter what is happening outside or within the space, what we've seen, and this is a publication, and again, any of these publications I'm happy to share with any of you, but across the board with our installations, before installation or before control of this variable, we saw a statistically significant increase in blast conversion rate, implantation rate, ongoing pregnancy rate, and a statistically significant decrease in our miscarriage rate, our loss rate. This is a very large study across multiple sites, over 5,000 cycles, 47,000 embryos, and 24,000 blastocysts. And this is the delta difference that we're seeing across the board with many of our installations. And again, everyone does IVF differently, as they should, but really the only thing they have in common is the removal of this variable. So this data has also driven us to launch into a very significant study. We've now learned which VOCs and what concentration of VOCs is most typical to the IVF laboratory. Knowing that, we were interested in how are they going from the air phase or the gaseous phase to the oil overlay, to the cell culture media, and into the embryo. How are they exerting those cytotoxic effects? And this is ongoing research to this day. It's fascinating. So just as that is a backdrop for our discussion, what I'd really like to talk about is what is the role of ambient air quality in pre-implantation toxicology? What are some of the common sources that we've learned and concentration of airborne pathogens in the typical IVF laboratory? What are the mechanisms of travel? Again, from air, the gaseous phase, to the liquid phase, to the cellular phase. And then once cellular, what are the mechanisms of cytotoxicity? And finally, I was asked to share with you the data from a very recent and large study conducted with a leading healthcare provider in our area relative to the impact of the technology towards, in healthcare, towards improved clinical and economic metrics. So to begin, I'm sure everyone is well aware, there are 3 categories of air contamination. Your VOCs, your volatile organic compounds, your viables, your biologicals, virals, fungal pathogens, and then your non-viables. These are the ones that are going to classify your clean room rating. And these are non-infectious and they're not cytotoxic to the embryo. However, they need to be controlled because they can serve as vehicles on which the infectious pathogens can attach and therefore stay viable and stay active within your air changes per hour within your space. So again, not cytotoxic, but need to be controlled. Wherever you are located, United States, throughout the world, we all have 4 sources of cytotoxic airborne pathogens that can enter our laboratories. The outside air, your recirculated air within the space based on what you're doing within the IVF laboratory, the fabric of the laboratory, the materials, and the air provided by the HVAC system itself. If we think about the outside air, construction, and again, this is one that impacted us over and over again. And I personally walked around the rooftop and hand selected the air intake for our laboratory. And for the first 6 months, it was a great location. But then again, a progressive hospital, construction, a road resurfacing, medevac traffic. So with construction and disturbed soil, cytotoxic or embryotoxic ketones and aldehydes and benzenes and alkenes are commonly generated. When you cut through pressed particle board or cut through wood, just for construction, cytotoxic formaldehyde, acetone, methylene chloride produced. Fresh asphalt, this is another one that impacted us. It's one of the greatest sources of embryotoxic toluene. Just general traffic exhaust, sulfur dioxide, formaldehyde, ethylene, toluene, whether it be from patient cars, general traffic, medevac traffic. This was a constant source. Restaurants and cafeterias. We often spend very long hours in the IVF laboratory. When you leave the laboratory, if you're smelling, something from a restaurant or the hospital cafeteria, that's actually burning fat, and that's releasing polyaromatic hydrocarbons and particulates. Again, toxic to the cell culture environment. Generators typically need to be exercised weekly, whether it be a hospital generator or a business generator. This exercised near our air intake every Tuesday, releasing alkenes, benzenes, toluenes, methanes, formaldehydes. Again, outside source air is extremely dynamic and very difficult to control. The second source, the air within your IVF laboratory. I'm sure all of you have opened up a sleeve of sterile tissue cultureware, and you can smell. You can smell the styrenes and the toluenes and the acetones. That's parts per million. Our data showed it was parts per billion that impacted our outcomes repeatedly. Your laboratory plastics, your gloves, your SOPs are going to generate VOCs, disinfection SOPs. And again, a lot of this cannot be avoided. Your equipment's going to off-gas. We would typically try to order our equipment as early as we could, plug it in, let it run outside the laboratory so that it was off-gassing outside of our laboratory space. We were even cognizant of when we replaced gasketing. We tried to take care of our equipment ourselves, and we tried to replacing gasketing in the incubators. We'd actually try to off-gas that gasket before we brought it into the laboratory. Equipment, gasketing, repairs can allow the off-gassing of cytotoxic VOCs for months, if not to years. Your own refrigerators, your freezers, as they cycle to maintain the temperature, can release VOCs. The medical gases supporting the incubators have a large number of VOCs, so inline filtration is key. And then we shed personal bioburden, the clinical staff, the laboratory staff, the patients. This was a publication. It wasn't specific to an embryologist, but it was specific to someone in hospital scrubs, sitting still, mask, hat, booties, and they actually monitored the amount of VOCs that were shed by this individual, as well as biologicals, fungal spores, and non-viable particulates. In fact, the individual sat still during the testing, and then they did a second round of testing where the individual was chatting, was talking, and with the rubbing of the cheeks against the mask, produced an additional loading of the non-viable particulates. So it was an interesting study. Third, the materials that you use in your laboratory. If you're thinking about expanding or building new, be very, very cognizant of the type of materials you're using, your ceilings, your walls, your flooring, adhesives, avoid at all costs. Paint, white. The greater the tint of the paint, the greater the off-gassing of VOCs. Caulking, sealants. And then finally, the air provided by the HVAC system itself. Again, we create the perfect environment for growth. Perfect temperature, perfect humidity. So therefore, the pathogens, the allergens, and the toxins that are in the air going through the ductwork, serving your laboratory, can continue to grow. This is an image taken by my colleague. This is downstream of the final HEPA filter serving the IVF laboratory, downstream. So there is nothing between this air and the laboratory. And if you look at this effervescent glow here, that's Aspergillus. That's a fungal spore. And again, downstream of their final filter. Why is this of a concern? Because fungal spores are a great source of fungal VOCs. And this is a short list, all cytotoxic. So it's critical to remediate your fungal microbial spores. Now, in addition to fungal VOCs that are common to the IVF laboratory can be polar, your isopropanols, your ethanols, your aldehydes, Non-polar, your benzenes, toluenes, styrenes, hexanes. Low molecular weight, high molecular weight, again, fungal, microbial, and biogenic. Biogenic are typically from shrubs, trees, and one of the reasons a fair number of programs in the United States at least have seasonal fluctuations in their clinical outcomes. Again, if you're able to smell anything right now where you are, that's parts per million. That's the sensitivity of our own system. Parts per billion is what is cytotoxic to the embryo. So when we had the drop, and then finally it took years because we were in a clean room, so we didn't look at the air. But once we understood that toluene was one of the factors impacting our success, our immediate thought, well, let's add more carbon. Let's add more potassium permanganate. That was not successful. And again, you don't know what you don't know. These were the learnings. Carbon does a great job and it favors the non-polar high molecular weight VOCs. However, it is not going to favor this list of the remaining 5, your polars, low molecular weights, fungal, microbial, and biogenic. Potassium permanganate does a great job of oxidizing or favoring some of the polar and low molecular weight VOCs, but again, will not remediate all. So what we learned is that filtration physics beyond carbon and beyond potassium permanganate were necessary to completely remove the embryotoxic VOCs. There are close to 90,000 VOCs. And again, carbon's going to remediate one biochemical or several biochemical families, potassium permanganate others, but there are thousands of others that will not be favored by either of those two media. Another approach that we had, which many do, which is not wrong, culture under oil. Use tabletop incubators, time-lapse imaging, anything that you can do to create a more closed environment, assuming that that will mitigate the entry of VOCs into the cell culture media. And this is not always the case. Let me explain. So if you think of each VOC, and again, they're up to 90,000, each VOC has its own unique fingerprint. The action of each VOC is dictated by a unique partition coefficient. In other words, its ability to enter cell culture media, its ability to enter the oil overlay, perhaps its ability to be biphasic and enter both. So each VOC has its own biochemical fingerprint that is going to dictate its ability to enter the cell culture media, the oil, by its partition coefficient. It's dictated by its level of hydrophobicity and vapor pressure. As I said, some VOCs are biphasic, and entry into the media or the oil can happen within seconds and minutes. Once present in the media, the VOC can then enter the cellular tissue or the embryo and thus remain a permanent threat to that cell. And again, this is part of ongoing research as we identify the most common VOCs and their concentration found in IVF laboratories, those most common VOCs, and then by laws of physics, how they enter the cell culture media, and then again by laws of physics, how they enter the embryo itself. For example, ethanols, acrylins, formaldehydes, glutaraldehydes, very hydrophilic, very water soluble, and therefore are going to rapidly enter your cell culture media. On the other hand, styrenes and benzenes are very hydrophobic. So they're very highly oil soluble and will rapidly enter that oil overlay. Once cellular, there are a number of outstanding publications, and again, I'm happy to share these, that have demonstrated the toxicity or the mechanisms of toxicity that once present, the VOCs have on the mammalian cell, and some of these are specific to the human embryo, by perturbing the cellular membrane, allowing leakage and disturbed homeostasis, increased intracellular calcium leading to cell injury and death, impaired mitochondrial function, spindle formation, chromosome alignment, reactive oxidative species overproduction, cross-linking of polypeptides, formaldehydes, glutaraldehydes, perturbation of DNA and RNA replication, increased DNA oxidation damage, and exerting an influence on epigenetic variation. So another, again, approach that we took and question that we're asked is, does adding a HEPA filter or an ALPA filter provide adequate or comprehensive protection of our culture environment? Again, from our own learnings, the answer was no, it is not providing comprehensive protection, and this is why. We had ALPA filters as our final layer of filtration, bathed in UV light. Again, we create the perfect environment for growth. This is what we're trying to do in that space. So as a random QC, I personally swabbed the ALPA filters above our IVF laboratory and was shocked when we got the report back from the micro lab that we had bacillus growing on our ALPA filters on top of our ALPA filters. So again, to put this in perspective, your HEPA filters or your ALPA filters are going to capture organics based upon size. So as they capture the biologicals, those biologicals are not killed. They will continue to grow. They need two things, food and water. Food is often the other organics, or depending upon the filter, it could be the matrix itself. Water is simply the humidity in the air. So you're capturing these organics above the space you're trying to protect, and they're continuing to grow. With high velocity going through those filters, they have the potential of entering your space. In addition, a lot of your smaller bacteria and smaller viruses are not going to be 100% captured by these final filters, and all of your VOCs are going to go through the media. So just to summarize where we are relative to IVF, the air serving our culture environment is extremely dynamic and influenced by the outside source air, all of the activities of your neighbors that are exceptionally difficult to control, the recirculated air within your laboratory or your clinical procedure rooms, the materials that are used, your protocols, your staff, and the HVAC system itself. These were all learnings from our own experience, and we've learned that many share these. We also learned that subtle, very low levels of airborne VOCs and biological contaminants are impactful to successful and consistent human embryogenesis and clinical outcomes. Embryotoxic VOCs in the gas phase can rapidly enter the cell culture media and embryo, thereby exerting highly cytotoxic effects. So in designing and using ultraviolet genomic and mathematical modeling and a proprietary engineered molecular media, our goal was to control this variable and drive it to zero. So the technology was designed to completely capture and inactivate all chemical and biological contaminants. And finally, it's critical to control the quality of the ambient air serving your culture environment to optimize and provide consistency of successful pre-implantation embryogenesis. Now, in shifting gears slightly, because the LifeAire systems technology was selfishly designed to protect the human embryo, the bar was set exceptionally high. We sought to design a technology that would deliver below detection total VOCs, kill of the anthrax spore. The anthrax spore is the most difficult spore to kill. We wanted a system that would deliver zero byproducts, no ozone, no intermediate molecules. We didn't want a system that would use catalyst, because catalysts degrade over time. We wanted the effectiveness of the delivery for the long term of the use of the technology. And we wanted a single pass delivery. In other words, whatever enters our system does not exit. It takes one time through the system, not 10, 15, 20 circulations. So with this as a backdrop, we've recently placed our technology in healthcare. In the United States, at any time, 1 out of every 20 patients will have an infectious complication acquired through the hospital environment, either via direct surface contact or airborne pathogens. It represents a financial crisis of $35 to $88 billion annually to U.S. hospitals. This is key. This is from the CDC. 69 to 80% of the pathogens that cause secondary and subclinical infections are airborne. Greater than 90% of all surface fomites on the surgical tray table, on the bed support, on the bed tray, greater than 90% of all surface fomites originate from the air. Those two key pieces are not well understood. Conventional HEPA filtration does not provide adequate remediation, and infection control protocols are not eliminating the problem. We've spoken with a number of leading hospitals in the United States, and everyone is doing a phenomenal job towards infection control and developing new protocols, but we're still not at zero. So our technology was designed again, to provide a kill of the anthrax spore. The system was actually tested by National Homeland Security Research Center. So the data that I share relative to the anthrax spore is coming from this testing data. If you're able to provide a kill of the anthrax spore, you provide a 9-log reduction of the airborne biological infectious pathogens that are of greater concern to us: MRSA, C. diff, Pseudomonas, Legionella, Aspergillus, TB, staph, strep, etc. And this is just a short list. On the viral side, we provide a 100% kill of some of the more common and infectious viruses. And if you see, a number of these are quite small. Your HEPA filter is optimal at capturing viral particulates greater than 0.3 microns. It will capture some down to 0.1, but it's not at 100%. So your influenza virus is 0.098 microns in aero diameter. SARS 0.13, the current SARS-CoV-2 is 0.12. So again, a high likelihood of penetrating HEPA filtration. So knowing that the system provides the kill of anthrax and a 9-log kill of your more common healthcare associated infectious airborne pathogens, we conducted a very large study, over 8,000 patients, with a leading healthcare provider in the United States. The goal of the data was threefold: environmental testing on a monthly basis, retrospective analysis of the clinical outcome data, as well as the economic data. So there were 2 floors as the layout. There were 2 floors identical. They were medical surgical floors. So the majority of these patients are surgical patients. In Zone A, these 16 rooms were completely protected by the LifeAire system. So all of their supply air and all of their return air, all of the air in these 16 patient rooms was provided by our technology. The second half of the floor, which was the second half of the 16 rooms, 32 in total, served as a control against Zone A. So they were pure HEPA filtration, your typical gold standard, but then did benefit from the return A coming from the LifeAire protected zone. And then our pure control, if you will, was the entire floor beneath that. Again, the study was, these 2 floors were chosen because they had an identical physical and clinical footprint, similar patient etiology, similar staffing, similar SOPs. There were no new initiatives that occurred during the study period. It was a 12-month study period. The study was blinded. It was double blinded to both the staff and the patients, so there'd be no subtle change in behavior. And again, we did prospective environmental testing on a monthly basis, and retrospective clinical and economic data was collected. if you think about what airborne pathogens are responsible for some of the more common illnesses in a med-surg unit, this is a short list of some of the bacteria and viruses that are each responsible for some of your more common illnesses. So our goal, completely remove these from those patient rooms, those 16 rooms. Just completely remove the airborne loading and therefore reduce surface loading. So the unit was installed. We went live January 1, 2018. 12 months later, at 8,255 patients between the two floors. Full capacity from the very launch of the study. Again, monthly, we did extremely aggressive and comprehensive testing of all airborne bacterial and fungal loading in several of the patient rooms in each of the 3 zones. Pulled over, this is called an Anderson pump impaction sampler, where air is pulled over a blood agar plate. We selected multiple blood agar plates so that we were capturing a vast array of bacteria and fungi. We also did surface swabbing in each of these rooms per zone. We were told which pieces of equipment, which areas were the most commonly touched clinical surfaces, which were the most commonly touched patient surfaces, and then we chose to also swab the final diffuser and final return. Again, I'm happy to share, and all of this is now published, so I'm happy to share the publications with you and go into far greater detail. But if you look at the control floor, again, very typical hospital flora. They had outstanding cleaning regimens, but this is very typical. If you look at the viable fungi by air, viable fungi by surface, you see a very similar pattern here. What's in the air lands on the surface. Viable bacteria by air, viable bacteria by surface. Again, relatively normal hospital flora, but each of these is associated with causing a patient illness. If you compare that to the pure LifeAire zone, there was 0 viable fungi by air and viable bacteria by air. We did have some viable bacteria on the surface. It was staph, and it was on the IV control plate and on the patient remote, so probably touch. But again, the most important, over testing monthly for 12 months, 0 viable fungi by air, 0 viable bacteria by air. So to summarize the study, and again, we're happy to share these publications, by completely removing all infectious airborne bacteria, fungi, and viruses, we saw a statistically significant reduction in infectious surface pathogens. So those 16 patient rooms had a removal or a reduction of airborne and surface vectors, if you will, of illness and infection. What did that mean clinically? Those rooms protected by the technology actually demonstrated a 39.5% reduction in length of stay. So there was an increased capacity, increased use of that space, improved discharge outcomes with those patients, and improved economics. There was a significant cost savings associated with those rooms. Beyond illness and infection, just facility readiness. And unfortunately, we've all experienced this time in history, these last 12 months. And after the study was completed, to their credit, the hospital network installed a second system so that all 32 rooms were protected, obviously from infectious bacteria and fungi, but also thinking about SARS-CoV-2. So they would have an isolated and dedicated floor specific for that protection. And again, this was right before they installed the second system on the second half of the floor. So I'm happy, Lars and I are happy to address any of your questions, and we thank you for your time. I know everyone's had a large number of virtual Zooms and presentations, and again, we just thank you for your time and look forward to continuing our discussions.
Okay, Thank you so much, Katie. We will answer any questions that you have to put into the QA panel. And if we don't have the time to answer them during the remaining time, we will get back to you and answer it. Use our website or the emails. So we have one question here. Let's see. We have just built a new lab with HEPA. I think we answered this one. HEPA combined with charcoal and kalium permanganate. Is that enough? If not, why?
Yeah, I think, and that's exactly what we did. I think, you know, when you think about carbon and potassium permanganate, again, they do a great job at certain biochemical families of VOCs, but there are thousands of others that are cytotoxic that are not favored by those two particular media. And HEPA, again, HEPA is a capture model, so it's going to capture your organics, and then they can be allowed to continue to grow. And obviously, all of your VOCs go right through a HEPA filter.
Okay, thank you. Another question. Let's see. Is it possible to fit your products into the existing ventilation system? How?
It's a good question. Lars, about half of our installations are retrofits, as we call them. So they're placed into an existing space or existing HVAC system. And what the process is, our engineers review the as-built, we call them the as-built drawings. Our engineers review those and assess your existing air handler, your ductwork, the cubic volume of air that you're trying to protect. And then basically our engineers will say, this is the size system to deliver the air changes per hour, meet your code, deliver the positive pressure, and protect that size space. This is the size system, and this is the best location for it to keep it as seamless as possible.
Yeah, totally agree. Let's see. I think we answered this one also. How effective is HEPA filtration? I think we covered that, but you can take it again then.
Again, HEPA does a great job of capturing organics of a certain size, and it's great for certain applications, but for in particular for human IVF, your VOCs are going to pass right through and a lot of some of your smaller bacteria and viruses. And that ability is going to impact, your healthcare environment, your patient setting.
Okay, thank you. Let's see, we have one more here. I think you have answered this also. How does the LifeAire system compare to the other HVAC systems in removing and remediating COVID?
So, Lars, the ability of any technology to kill SARS-CoV-2, or really any bacteria or virus, it's based upon the genomic sequence of that bacteria and virus. And so we know, because our technology was designed to kill the anthrax spore, we know based upon the genomic sequence of the SARS virus that our technology provides a 145 log kill of COVID-19, the existing technology system, and 6 log of sterility. So it's an exceptionally high kill. The other thing that's unique is that our technology is modeled specifically for your metrics. So it's modeled based upon your space, your ductwork, your air velocity, so that this kill is guaranteed, that the kill is going to be delivered. And finally, a single pass. We wanted, if there's SARS-CoV-2 entering on one side, we want to know it's completely remediated on a single pass through the system.
And no byproducts?
No byproducts, no.
Okay, all right. So finally, I think this is the final question. Again, remember, you can send questions to us, but we will also have a video of this webinar that we will send out shortly, I hope, to all the participants. So the final one, I think it's also been answered. How does the LifeAire system help patients in healthcare settings? And especially that I'm come to think of elderly homes, etc., etc. There's so many possibilities.
Mm-hmm. Yeah, I think, you know, our goal with the large clinical study was that our technology would completely remove all airborne bacteria, viruses, and fungi. And by doing so, dramatically reduce surface loading of infectious pathogens. So by doing both of those, you're removing two very common vectors of illness for those patients. And that's what generated that. The result of that was the dramatic decrease in length of stay. Now, length of stay is a key economic driver for hospitals. And so that's what then generated the $2.3 million savings specific to those patient rooms in a 12-month period.
Yeah. I have one here also. What do you think about systems offering photocatalytic oxidation to minimize VOCs?
So when we were designing the LifeAire system, we looked at a lot of technologies to incorporate into the system. And we did look at PCO. The concern we had with PCO was the potential use of catalyst. And catalyst, I mean, they can perform very well initially, but catalyst can degrade over time. And so it was important to us that our system not have something that was going to degrade over time so that we could guarantee the effectiveness, you know, beyond the point in time when the replacements had to occur.
Yeah, thank you so much, Katie, and everyone that was here in the webinar. And we will answer all the questions. And as I said again, we will send a video out to all that has not only checked in, but also those that previously wanted to see, because I've seen from some things here that they had problem with linking in. So we just have to see that you get them as the video as soon as possible. And again, you can use these emails that you have here on the screen to contact us for more information. And also seeing that you want to have the publication, and it's I can send that, and then I think it was Ronnie Jensen. I can send that to you. So that will be fantastic. So thank you all again. And I see now it's coming in here from different parts of the world. And as I said in the beginning, those that weren't here, we were represented from the whole world and most of the different societies as well. So thank you so much for bearing with us. And again, thank you and take care.
Thank you. And everyone stay safe.
Yeah, the same.
Thank you, Lars.